Thank you, Dr. Chen. So thank you for introducing me, and this is my presentation. I know this may look like a weird jumble of words to some of you, but I promise I will explain it further. Um, so first, this is Papa Jack, as Dr. Chan mentioned earlier. He is pretty much one of the sole reasons research was founded here, as it was his dream to have students be able to conduct research and allow them the resources to conduct summer projects. And that's how the K Fellowship kind of got started. So thank you so much, and we'll also do more acknowledgements later. So first, my project is centered around native Hawaiian snails. So native Hawaiian land snails have achieved great diversity because of their adaptation in isolation. So this is essentially saying because we live on an island that's so isolated from, for example, the mainland or Japan, right? We just got lots of ocean around us. Um, there is a lot of endemic species here. Specifically, there is actually over 600 endemic species of snails. And these are some photos of some of the really pretty ones. And this is actually one of my favorite ones because they have a really unique shell coloring and they're very pretty to look at because they come in lots of different ranges and they have lots of different patterns. Next, Hawaiian land snails also provide key ecosystem services. So traditionally in native Hawaiian, like a pure native Hawaiian environment, we actually didn't have earthworms till about like the 1800s, I think. So actually the role of the earthworms was pretty much completely filled by these snails, as these snails were responsible for the turnover of decaying matter, as well as cleaning basically fungus and microbes off of leaves, as you can see in this photo. And by doing this and cleaning off this debris kind of on top of the leaves, usually of native Hawaiian understory plants, it allows photosynthesis to be achieved at a higher rate, which actually help them grow. Next, in Hawaiian customs, snails were known as the voice of the forest and embodied the passing of one's genealogy through singing. So, this is just one example of how snails are really important, not only ecologically, but also culturally, as there's so many of them and they are very prevalent um, in the native Hawaiian ecosystem, as once there were so many that like on the palms or the fronds of trees, when like the wind blew and knocked the fronds together, then you could hear like the tinkling of the shells hitting against each other. And it also they are also literally the sound of the forest, according to some sources, because they kind of represent passing the oral tradition of passing on genealogy. Next, unfortunately, most native Hawaiian snails are endangered or already extinct. So this is why my project focuses more on the conservation of these species. So this is the genus I'm working with, which is the succineid. And this is more examples of the pretty and unique patterns that exist on the snails. Next, in order to basically conserve the species, I'm exploring something called captive rearing. So captive rearing is used to prevent the imminent extinction of endangered species. So on the left is a little bit more of a basic captive rearing setup where you basically have like a little micro habitat or environment for the snails to live in. And it's kept usually in like a plastic container. And on the right is a little bit more of an advanced version where there's like day and night cycling and there's water, like rain simulations. My project deals more with the left, but there's a wide variety of captive rearing setups. Additionally, captive rearing includes dealing with the snail's microhabitat and its expelling of waste, such as ammonia, which we find later in the presentation to be actually pretty interesting because a lot of the nitrogen cycle is important in understanding our process or our results. But the important thing you have to understand is not ammonification, but this nitrifying bacteria and the role that they play in basically converting nitrogen from one form to another. Additionally, recent mortality events in captive rearing have prompted research on snail diet. So actually locally, I think there was one organization that had a captive rearing setup, but they also encountered a lot of mortality events where a bunch of snails died either due to pathogens or maybe not a good enough diet. So there was that prompted like a research on the snail's diet and how that could lead to improved fitness. So that kind of gives some background on what I want to try to do. To give more background on the microbiome, it's basically the collection of microbes. So that would be all of your bacteria, all of your fungi, and all of your archaea. Basically any living thing. Like, for example, we hear it a lot when it's in reference to humans. For example, the gut microbiome of humans. But there's actually a lot of different microbiomes, as there's a microbiome of like your skin, for instance, and there's microbiomes in many different plants and animals. Next, according to some of the research papers I've read, more research is needed to explore how the microbiome assemblages influence snail indicators of health, such as fecundity and survivorship. So fecundity is basically its ability to reproduce or lay eggs in this case. Some research studies have used proxy species in lieu of the real ones due to moral concerns and endangered statuses. Because this is kind of a relatively new setup, in our experiment, we used invasive species to kind of stand in for the native species just in case we killed some of the snails, which you'll learn more about later. So this funnels down into my three main research questions which was, does the microbiome of Succinea unicolor change over time in captive rearing? And can the microbiome change be an indicator of snail survivorship? And is there anything we can learn about how to better rear any of the Succinea species, specifically the native ones? So then we get into my methodology or how I tested this. So essentially, I collected snails from the wild, and that's a picture of Sumida Farms. They allowed us to grab 15 adult snails, and essentially I had them in little micro containers, and I basically waited until they pooped um, to get DNA out of them. But after they pooped, I moved the snails into basically groups of three into small containers, and that was their captive rearing setup. But this is to get the microbiome of what we would consider their wild habitat. So we extracted all of the different poop samples and we combined them, and then we ended up sequencing them, which I'll talk more about later. Again, at time two and time three, we did similar things where since they were already in their little habitat, we took like three random poop samples from the habitat. As you can see pictured on the left, that's what it looks like in real life. And I collected them all into a sample, and then I ended up extracting the DNA from that and sequencing it. So to go into the sequencing results. First, this is a principal coordinate analysis or a PCOA of the community replicates through time. So I know it looks really confusing right now, but I will break it down in the next slides. First, I discovered that the microbiome does change over time. So in this first graph, we see that all the boxes at time one generally look the same because they're kind of clustered together. But then at time two, they spread a little farther apart. And then at time three, they're really different from one another. Therefore, we know that the microbiome changed from the first time point, which was this one, to the last one was quite different. So now we know that the microbiome does change over time. Next, the microbiome change for each replicate is not exactly the same. So, um, okay. Um, the microbiome change for each replicate is not the exact same. So this is the microbiome change for box two. So you can see here, there's kind of clustering down here, but then at time point three, it travels a big distance, which means it traveled like, or it changed over that time point. This is another box. This is another box. This is another box. And this is the final box. So they all change kind of in different directions. So you know that although all of them changed, they may have changed in slightly different ways. So to break it down further, I'm going to show you guys a relative abundance chart. So it may look kind of intimidating, but basically all it's saying is whatever colors are here, which are represented here, make up whatever's in that sample. So For here, I have 5 boxes: box one, box two, box three, box four, box five, which were like the 3 box communities or the 5 box communities that I had. And they all generally right now look the same, which makes sense with the clustering pattern we saw before. Next, they get a little different, but generally still around the same level of content, which also makes sense with the general clustering patterns that we saw earlier. And finally, we have time 3, where we see distinct changes, especially in boxes 1, 3, and box 5. As you can see, there's a presence of cyanobacteria, which is like the seafoam green color. Additionally, this blue color right here definitely takes over box 2 and box 4, as there's very little of the yellow color left. Um, so from time 1 to time 3, the cyanobacteria became prevalent in boxes which actually had all of their snails die. So from between time points 2 and time points 3, we had a lot of our snails die in captivity. Pretty much all the snails died in all the boxes except box 2, which is interesting because we see that in box 1, box 3, and box 5, where all the snails died, there was a high presence of cyanobacteria, which we did not really see before. Additionally, ammonia is the inorganic source of nitrogen preferentially used by most cyanobacteria. So ammonia is produced naturally from decomposition of organic matter, such as animal waste and plant matter. And this is interesting because we know that since cyanobacteria might prefer ammonia, then ammonia might be in the boxes, because we think that cyanobacteria could be used as an indicator species, because the high presence of cyanobacteria within these boxes would show that ammonia ended up being present. And additionally, succinaids do not process waste similarly to other snails, as they do not make as much uric acid, and instead directly expel waste into their environment in the form of ammonia. So uric acid is this thing right here, and ammonia is this thing. And essentially, they're both wastes and they're both toxic, but ammonia, which is this one, is a lot more toxic to the snails, I think. And this was what was expelled by the succinaids, because according to biology for this particular genus, they tend to expel more of this than they do the uric acid, which is considered less harmful. Therefore, if we circle back to my beginning questions, does the microbiome of Succinea unicolor change over time? Can the microbiome change be an indicator of snail survivorship? And is there anything we can learn about how to better rear the Succinea species? So yes, we know that the microbiome does change over time, as shown by our graphs. And next, can the microbiome change be an indicator of snail survivorship? So in this case, we won't really know for sure, but we can at least say that the cyanobacteria present um, in boxes 1, 3, and 5 was interesting, because all of those snails ended up dying, and cyanobacteria is an indicator species of ammonia present. So it would be possible that ammonia ended up basically accumulating in these boxes and making the snails die, but we would have to do further research to really find out if that's the case. Additionally, is there anything we can learn about how to better rear the Succinea species? So, in regards to this, I think over my process, at least, I learned a lot about how often you have to clean out their waste, especially because I noticed that their boxes got very decayed very fast. So I think that planning a environment that doesn't decay as quickly, as well as letting their kind of gases off breathe into the air and maybe making bigger boxes, would be a good change to rear harder Succinea species. Research on the biology of the family Succineidae in Hawaii may give insight into a foundation for conservation management and strategies. This is just an example of what further research could do for the more endangered families that live on our islands. Finally, I'm getting to the end, I promise. My reflection. Basically, I'm supposed to answer 2 questions here. What did I learn, and what advice would I give to somebody who wanted to pursue this in the future, K fellowship or research in general? My number one piece of like advice, or what I learned, was ask questions if you're confused, because nobody will like be like, oh, why don't you know that, you know? And everybody wants to help you learn and grow. And I feel like asking questions is the best way to learn because you definitely clear up a lot of like misconceptions in your head, especially because there's a lot of jargon and terminology when you research that you may not be familiar with before going into it. Next, don't get discouraged by obstacles. So, fun fact, I killed, no, the snails, 60 of my snails died in the process of this experiment. So, don't get discouraged by obstacles. And as Mr. Hill says, trust in the process and you will get results. Finally, my acknowledgements. So first is my mentor. This is Jan. She's also online. And she works as a malacologist in Bishop Museum. Along with the whole Bishop Museum team, they were really instrumental in me conducting my research, and I wouldn't be able to get where I am without them. For example, Dr. Hayes and Dr. Young, they were really important, especially in consulting my experimental design. As long as all the staff at the malacology lab and PCMB, they were really, really helpful and supportive while I stayed in the corner of their lab for like half the summer. Sorry. Thanks for buying me lunch and stuff. Um, but also the Iolani Wet Lab and Iolani staff, the Iolani Community Science Department, all of the teachers, Dr. Chan, Mr. Hill, Ms. Kobayashi, Dr. Perez David. You guys were all such amazing help over the summer, especially during extractions. Thank you. Um, and yeah, my parents and definitely the fellow K fellowship people were really helpful in providing moral support and driving me places. So it was definitely a team effort. And yeah, thank you.